Right. Homework. I gave you homework in the last lesson, so we're gonna revise it now. If you didn't do the homework, then I suggest that you go to the previous clip and then do the homework first. We don't want you wasting your time here and your data. Huh? Right, so this was your homework. November that. Give me a second here. Forgive me, I just switched on my computer. So there might be a bit of hiccups. Right. Let's leave it like that. Right. Let's go to our homework. November final 2018, question 1.18 and 1.19. I hope you can see this side. Because I want to put them there. Okay, what should I do? I need the board. So let's move this there. Those are the questions. Let's put the smart board that side. Beautiful. I'm assuming that you have the paper in front of you. I hope you can see. See if we can try to make it a bit bigger. Nope. Can't do. This is the best. Right. In a certain species of rabbits, body color is controlled by what? By two alleles. Remember what we said? We understand the question first before we even think of attempting it. So body color characteristic is controlled by two alleles, where black is dominant to what? To white. And then there is also what? So there is one characteristic here, body color. There is another characteristic here, what? Ear shape. And in ear shape, so in body color, so we have body color. You need to break down the question. So for you to understand, in body color, black is dominant to what? To white. And we have another characteristic that we're coming up with, ear shape. So ear shape, and in ears, white ears are dominant over narrow ears. What is the possible genotype for what? A black rabbit. For you to get this phenotype of black, because it is what? It is the dominant one, we're going to have to find it either like this, or it's going to be like this. Either homozygous dominant or heterozygous. And the black rabbit is what? Has narrow ears. And from what we know here, we know that this is a recessive allele. So if it is a recessive allele, only one way we can find it in our genotype, it has to be this. So in other words, what is the possible genotype for a black rabbit with narrow ears? Then the possible genotypes are either this for black, this for narrow ears, this for black, and this for narrow ears. Then I go and check my questions. Which ones? By a process of elimination, A is out. Why is A out? Because it is giving us what? White ears. Anything that doesn't have this is out. Because we're looking for a black rabbit with narrow ears. So this is also out. Our two options are these, B and D. And which one matches any one of these? It is D. So D was the answer. Right. What is the possible genotype of gametes produced by a white rabbit? And we know that white is what? Is recessive. So every answer has to have this. Every answer. Anything. Right, anyway, that is a white rabbit. With narrow what? With narrow ears. Narrow ears is this one recessive, right? Then. But what are they asking for? I thought they're asking for the genotype. For a full complement of a genotype. But look at this. What is the possible genotype of gametes produced? If meiosis occurs here, we can only get one type, this and this together, this and that together, this and this together, this and this together. Same thing. Same WhatsApp group. We're only getting one type. So what is the type? Hope you get that. I hope you're getting that. I want to move a bit faster here so that we go. Right. November final. The next question was from here. 2017. Oops. Oh, there we go. This November final 2017. Let me open it up. Where is 2017 final? Good. And what are the questions that we're looking at? We are looking at 1.18 to 1.110 and 1.4. Let's start with 1.18 to 1.110. Right, there we go. Shall we remove this? Right. Let's continue. The diagram shows one pair of homologous chromosomes found in each of 4 fruit flies. The alleles for what? Antenna shape. This is a characteristic. Gives you what? So the alleles for antenna shape give you normal or abnormal. And then the alleles for body shape, body pattern, sorry, gives you an allele that gives you stripes and another one that gives you what? Without stripes. And these alleles are indicated on the chromosomes. So how many flies are we looking at? We're looking at 4 fruit flies. So what are they saying? The diagram shows one pair of homologous chromosomes found in each of the 4 fruit flies. So this is one pair. This is another pair. This is another pair. This is another pair. How many characteristics are we looking at? 2 characteristics. How many alleles? 4 alleles. Right. Because each characteristic is how many alleles? 2 alleles. And how many flies are we looking at? Fruit flies? 5. 4 of them. 1, 2, 3, 4. Right. Here's your key. Alright. Let's zoom it up. Let's scroll down a bit. Let's go to the questions. Which fly is homozygous? Which fly is homozygous? Homozygous. For antenna shape and heterozygous for body pattern. If it is homozygous, it has the same allele. Doesn't matter here. They're not asking you whether it is heterozygous or if it is homozygous dominant or homozygous recessive. They're just asking for homozygous. Which one is a homozygous for antenna shape? We have our key here. We need a fly that has this or this together or this together. For us to get what? Homozygous for antenna shape. Is this one homozygous? Yes. Is this one homozygous? Yes. What am I looking for? It? Antenna shape. Is fly 3 and fly 4 homozygous for antenna shape? No. So immediately these are out. Our answers are going to come from there. And then we haven't even finished the question here. And heterozygous for body pattern. This shows you body pattern. Body, an allele for body with with stripes and another for body without stripes. And if it is heterozygous, it means that it is consisting of what? Two different alleles. We are left with two options here because this is already cut out because of attenuation. Are these the same? Yes. Are these the same? No. What are they looking for? Heterozygous. So what is our answer? It's fly 2. Where is fly 2? The answer was C. Right. Let's continue. What is the genotype of fly 3? What is the genotype for fly 3? Let's remove this. Right. What is the genotype for fly 3? Where is fly 3? This. That. Because this is this, and it is the recessive. What is this? And what is that? Why am I starting with this one here? Because it is the dominant allele. And then we finish with the recessive allele. This was the genotype. Which answer gives us that? It is C. Let's continue. Which one of the following will represent the possible gamete for fly 4? What are we looking for? The possible what? Gamete. For which fly? Fly 4. We can't know the possible gametes for fly 4 up until we generate what? The genotype. Let's do the genotype. It has the dominant. So if I say fly 4 here, if I say fly 4, it has the dominant allele for normal antenna, and then it has the recessive, and then here it has the recessive body without stripes. What are the possible gametes? We generate them ourselves. This is what? Meiosis. This one and this one can be together in a gamete. This one and this one can be together in a gamete. This one and this one can be together in a gamete. And this one and this one can be together in a gamete. These are the possible gametes. So which one has that? Which one has that? Do we have this in our possible gametes? No. So it goes out. Let me scroll this up. Do we have this in our possible gamete? A. Sorry, wait here. 1.110. Do we have this part here in our possible gamete? Yes. Do we have this one? No. So A is out. Do we have this one? No. Why? Because it's showing only dominant alleles in one gamete. Do we have that there? No. Do we have this one here in our gamete? No, we don't have, because we don't have stripes. Do we have this? Yes. The answer was D. Right. And then the next question after that, I believe, was 1.4 in the same paper. Yes, beautiful. Same paper. Let's continue. Remove this first. 1.14. There we go. Mendel. Let's blow it up first. Mendel observed some characteristics of the pea plant Pisum sativa, which he suggested were controlled by inherited factors. He conducted a series of experiments in which he crossed pea plants He crossed pea plants. Oh gosh. Give me a second. He crossed pea plants with contrasting phenotypes. With what? Contrasting. Different phenotypes. To obtain the offspring of the F1 generation. At first, his crosses were simple and only involved what? One pair of characteristics. Mendel counted the number of offspring, showing each of the variations. So he did this. After he did that, what did he do? He counted the number of what? The number of offspring. That's what he was counting. And these results are shown in the table below. There they are. So what did he do? Can you see that he actually did monohybrid process? Where are you? Right. So he did his monohybrid process and then he got results. These were the results. I need to understand this table before I even go to the questions. For the characteristic of seeds, for the seed part of the plant, he looked at the characteristic of what? Texture and color. For the part of the pods, he looked at what? Texture and color. For flowers, he looked at flower color. And then for stem, he looked at what? Location of flower on stem and also height of stem. Hope that makes sense. Okay. So he did his crosses. In seed texture, he took rounded seeds with record seeds, and then he gave us our F1 generation. For seed color, he took those that had yellow seeds, crossed them with green seeds, and he got the offspring with all yellow. And he did the same for all the other characteristics. Right. Give the term for inherited factors. What are the inherited factors? Where are you? Only one thing here. That's genes. A cross involving only one characteristic, mono hybrid. Name the female structure of the flower where meiosis occurs. Ovary. You can write gynosium and all those, but ovary, yes, it's also correct. It's the most correct one. That's the one that's in your textbooks. Right. Use the information in the table above. Let's remove this. Let's remove this. Use the information in the table above to give what? The number of what? Of each characteristics of pods. How many types of characteristics on pods did we look at? We looked at pod texture and pod color. So what becomes our answer here? The answer was 2. Because we looked at 2 characteristics on pods. Pod texture and pod color. If I'm looking at the table, under pods, we look at texture, pod texture, and pod color. Right, let's continue. I hope you got that. Use the information that you're about to give the number of alleles for what? Seed characteristics. In other words, from the table above, How many alleles did we have for seed characteristics? How many alleles for seed characteristics? In other words, from this table, let's go back to the question. Give the number of alleles for seed characteristics. How many seed characteristics do we have? We have 2. We have texture and color. But how many alleles do we have? For seed texture, we have 2 alleles. One is for round and the other allele is for wrinkled. For seed color, we have yellow allele and a green allele. So how many alleles in total for seed characteristics? 4. That was your answer. Give the characteristic that is dominant. Give the characteristic that is dominant for flower color. So we go to flower color. So what do we see from Mendel's process? He crossed parents. One parent was violet, the other one was white, and guess what? All the offspring were what? Were all violet. So what is the dominant characteristic? It is violet. Right, walk with me. Give the characteristic that is recessive for stem height. Where is stem height? Here it is. Height of stem. He crossed the parent that was tall and the parent that was short, and guess what? All offspring were all tall. So which is the recessive characteristic? It is short. Why is it that the recessive characteristic? Because when he crossed tall and short, he only got tall. It means that the allele for short was masked. Complete dominance. If the individuals of the F1 generation are crossed, if the individuals of the F1 generation are crossed, oh gosh, sorry, give me a second here. There we go. If the individuals of the F1 generation are crossed, how many phenotypes of seed color will be expected in the F2 generation of seed color? How many what? How many phenotypes will be expected where? In the F2 phenotypes. Let's go to seed color. Where are you? When I'm looking at you, sorry, give me a second. If individuals of the F1 generation are crossed, how many phenotypes of seed color? Right. Sorry, there we go. There is seed color. We have yellow and green crossed from P1, and all the offspring F1 were yellow. But look, one parent was yellow, the other parent was green. And yet, when we crossed them, all the offspring were yellow, had yellow seeds. What does that tell us? That green is recessive to yellow. And in here, these F1 offspring are what? Are heterozygous. They have heterozygous genotype. Why? Because even though yellow and green gives us yellow, it means that the green is recessive. It's being masked by what? By the yellow. So the phenotype becomes yellow, but the genotype is, if yellow is dominant, then the genotype of the F1 is like this. I hope that makes sense. And if we cross, and if we allow cross, if we allow F1 generation with this, if the individuals of the F1 generation are crossed, how many phenotypes will we get? We're going to get 2. Why? Because this one will give us this one will give us that, this one will give us that, and this one will give us this. If you do this cross here, you get it in what ratio of dominant to recessive? 3:1, because all these are yellow, but this one is what? Green. So, how many phenotypes are we going to get in the F2? We're going to get 2. I hope you understood that. That is your answer. And we are done with this question. Let's remove this and go to the next one. I hope you're getting it. Genetics is easy, guys. Right. November 2016. Question 2.1 to 2.3. Let's go to 2016. There we go. Where is 2016? There we go. The leaf color in a plant. This seems to be relevant. The leaf color is controlled by what? 2 alleles, green and yellow. And then there's another characteristic. So this is a characteristic. Thorns on plant stems is another characteristic. So how many characteristics are we looking at? 2. Are controlled by 2 alleles again. Presence of thorns and having what? No thorns. 2 plants with this genotype were crossed, and then the offspring were then left to what? To pollinate each other. Their offspring. The table below shows the genotypes of what? Of the offspring of the second generation. Walk with me. 2 plants with the genotype this and this. So this was what? F1. Because they were crossed together, and then their offspring. Sorry, this is P1. And their offspring, what are their offspring? F1. Were then left to pollinate each other. The table below shows the possible genotypes of the offspring of this second generation. That's what? F2. Genotypes 1 and 2 have been left out. Yes, second. Right. So this is the course of what? The table below shows the possible genotypes of the offspring. So the offspring, these are the offspring. This is what? F2. Give the genotype of I. Where is I? I is here. So we know that the G goes there, and then this one here comes there. So what does it become? Cross with that one. Write it any other way, the answer is wrong. Give the phenotype of 2. This. This goes there, and this one comes down. What do we get? Oh gosh, I made a mistake. My bad, my bad, my bad, my bad, my bad. Mistake! Mistake, 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 mistake. Give me a second, give me a second, give me a second. I'm tired, guys. Forgive me. Give the genotype of I. This goes there, and this one goes there. So this, that, this, and that. We start with the dominant one. Yes. This is correct. I'm sorry. What did I tell the last one? I mixed up the alleles for different characteristics. What is the phenotype here? This comes here and this one crosses with that, giving you this. This is the genotype. Right. So we need to know what that is. Can you see how they are homozygous recessive for color and homozygous recessive for height? Oh, sorry, it's not height. Homozygous recessive for what? Leaf color. Homozygous recessive for what? Thorns on plant stems. So if this is like this, it gives us what? Yellow. And this gives us what? No thorns. So what was the phenotype? It was, give me a, What was the phenotype here? From this genotype. It was yellow. Yellow what? Yellow leaves with no thorns. Cheek here and a cheek there. Right. List the four. I hope you're getting it. Right. Oh, something is wrong with this one. I hate this whole minimizing and bringing it up. But anyway, it is what it is. Right. List the four genotypes of the offspring of the second generation that will be phenotypically different from the original pair. This one I just cleared it up. Right, let's do that. Where are you? There we go. Right. List the four genotypes. Again, I told you, I hope you have your questions next to you. List the four genotypes of the offspring of the second generation that will be phenotypically different from the original parents. The original parents are the grandparents. Remember something. What did you start with? You started with two plants with this genotype being crossed together. So they were crossed, they had offspring. And then the offspring were crossed again, and they gave us this. So it's like saying, I crossed with someone, and then I had kids. And then the kids grew up and had their own kids. I hope that makes sense. So the original parents, what did they have? One parent was this, and the other parent was that. Is that not so? What is this? This is green leaves with thorns. What is that? This is yellow. Yellow leaves with what? Without or with no thorns. These are the original parents. These ones here. Let's go to the question. List 4 genotypes of the offspring of the second generation. That would be phenotypically what? Different. If they are going to be different from this, what this means is that for these offspring, we are looking for offsprings, the genotypes of offspring from this cross, these offsprings. We are looking for what? We are looking for the genotypes that would make them to be different from this. Because these are our original parents. Now, if these are our original parents, for them, for the answer here, we are looking for something that is not like this. How many of the great F2 grandkids? They're not animals, they're not human beings. But you get what I mean. Remember, this was P1. So here, if there's a green leaves and with thorns, then we should have green leaves. But this time without thorns. Which genotypes give us this? For me to get green leaves, green is a what? It's a dominant. I'm either going to have this or I'm going to have this. Without thorns. Where's my without thorns? No thorns is what? Recessive. So these genotypes, these two genotypes, ensure that the offspring here are not going to be the same with any one of these parents. I hope it makes sense. And then here, this parent here had yellow leaves without thorns. Yellow leaves without what? Without thorns. So which offspring are going to be different? They are those with the yellow leaves. But this time with what? With thorns. With what? With thorns. Can you see how these types of phenotypes are not the same with the original phenotypes of the original parents? Yellow leaves with thorns. Yellow is recessive, therefore it needs to be homozygous, recessive. And with thorns, thorns is dominant allele, so it can be homozygous, dominant, or heterozygous. And how many marks was this question? It was 4 marks. So all you had to do was to state the 4. 1, 2, 3, 4. I hope you got that. Let's go to the next question. Ooh, where are you? Bingo. I like this question. I liked it. Let's do it. Right. The diagram below represents a phase in meiosis. Cell 1 undergoes cell division to give rise to two cells, cell 2 and cell 3. Some alleles are indicated by letters. Right. Explain why cell 1 does not belong to a human. How many chromosomes do we have in cell 1? We have, by the way, this is meiosis. How do we know that it is meiosis? Because each chromosome consists of two identical units called sister chromatids due to what? DNA replication, and these two chromatids are joined together at the central main. There. So we know that this is meiosis. And we know that it is meiosis 1. Why? Because they are found with their sister chromatids. And they're also messed up in homologous chromosome pairs. As a matter of fact, from term 1, we know that this is metaphase what? This is metaphase 1. Are we together? Right. Why? So how many cells? Why is this cell? Cell 1. Why does it not belong to a human being? Because in cell 1, how many chromosomes do you have? You have this chromosome 1, chromosome 2, chromosome 3, and chromosome 4. How many chromosomes do you find in a human being? 46 chromosomes. So this cell 1, does not belong to a human being because it has 4 chromosomes in its diploid state instead of having how many? 46 chromosomes. Or alternatively, this is a pair, a homologous chromosome pair. This is another homologous chromosome pair. You could also answer it looking at the context of pairs and say that cell 1 does not belong to a human being because it has 2 pairs. 1, 2. 2 pairs of homologous chromosomes instead of having how many? 23 pairs of homologous chromosomes as in a human being. This question comes because you guys, you think that every diagram that we give you of meiosis is of a human being. I don't know why. I don't know the mind of a student. But that's what you think. So we know that's what you think. Now you know the concept behind it. There are 4 chromosomes in here. And that's impossible in a human being. Because we have 46. Or there are 2 pairs of chromosomes, homologous chromosome pairs. And yet in a human being we know there are many? 23. Right. Let's continue. Where are you? There we go. Ooh. Right. How many chromosomes? How many chromosomes will be present in cell 2? How many chromosomes will be present in cell 2 at the end of telophase 1? At the end of telophase 1. So these chromosomes, this is metaphase. What is the next thing? This is metaphase 1. What is the next thing? Anaphase 1. And what do you end up with at the end of that? We have telophase, and they split. This cell splits into cell 2 and cell 3. So these go there, these go there. So how many chromosomes do we end up with in cell 2 at the end of telophase 1? We end up with 2. This is cell 2. How many chromosomes are coming this side? chromosome 1, chromosome 2. So we end up with 2 chromosomes. How many chromosomes will be present in the daughter cells? How many chromosomes would be present in the daughter cells produced by cell 3 after what? After meiosis 2. How many daughter cells? There are 2. Because bear in mind something. This cell and this cell are going to go to meiosis what? Meiosis 2. This one will go to meiosis 2, and then this one will produce 2 haploid gametes. This one will produce 2 haploid gametes. So, what is the question? How many chromosomes will be present in the daughter cells? How many will be present in the daughter cells? Right. How many chromosomes will be present in the daughter cells produced by cell 3? Where is cell 3? This is cell 3. So in other words, cell 3 ends up with this chromosome and this chromosome. After meiosis 1. Shall we draw it here? This is now cell what? Cell 3. So what do we have? We have and then we have and then this is after meiosis what? 1. And then it goes to meiosis 2. After meiosis 2, what do we have? We have a further 2 cells from cell 3. This one will end up in here. This one will end up in here. This one will end up in here. And this one will end up in here. Sorry. What is the question? How many chromosomes will be in the daughter cells? What is the answer? 2 chromosomes in each daughter cell. Draw a labeled diagram. Draw a labeled diagram Let's do it here. I'm going to do a quick sketch. A quick sketch. Because we don't have time. Draw a label diagram of a gamete that will result from cell 2. In cell 2, at the end of meiosis 1, you're going to have this one and this one in here. Are we together? So in other words, in cell 2, at the end of meiosis what? 1, you're going to have, I'm just doing a sketch here. That's what we're going to end up with all the alleles what? Dominant alleles. And at the end of meiosis 2, what are we going to end up with? This one, we're going to end up with 2 cells, haploid cells, gametes. This one will come in here. This one will come in here. This one will go in here. And this one will come in here. And what are they asking you for? Of only 1. So all this, we drew 2. But can you see that the 2 are the same? So they only want 1. So we give them the one that they want. That is the one. There we go. Every diagram in life sciences has to have what? Has to have a caption. Draw a label diagram of a gamete that will result from cell 2. So my caption might be diagram of a gamete produced by cell 2. I underline that. And then I have the diagram. Each diagram should be at least 6 lines to half a page. Not more than half a page. Are we together? It needs to be clear. That's what I'm trying to say. And then what do you write? And then you put your labels. Can I just go to the memo? Save us time. There. If you follow the instructions, they're asking you for what? For 1 gamete. Instead of what? Instead of 2. So you drawing a single cell is 1 mark. You drawing 1 cell instead of 2 cells is 1 mark. Only 2 unreplicated chromosomes are showing. Another mark. Short unreplicated indicating what? Dominant T. And another one indicating dominant R. And any one of the correct labels. Ladies and gentlemen, we don't draw a diagram without labels. So any one of those was a mark. I hope you got that. I hope you got that. Right. Right. What we do. State Mendel's law of segregation. State Mendel's law of segregation. Shall I just give it to you? There we go. A pair of alleles. Tick. The pair of alleles, tick, on homologous chromosomes. On what? Homologous. Homologous has to be there. On homologous chromosomes separate during meiosis, tick. So that only 1 allele of each pair is present in the gamete T. That is Mendel's law of segregation. Even the one I gave you, still fine, you will get your 3 marks. Right. Is there any other question that we haven't done? 2016. Let's go there and see. Look, we are done. The aim is for you not to pass but to excel. You can't excel and get distinctions if you haven't been able to get them in your classworks and homeworks. Hope you enjoyed the lesson. Goodbye.